All right, welcome everyone. We have a very exciting talk for you today. Today, Yoshie will be giving a talk on how to get started with Cryo-EM at CEMAS. So Yoshie has been at CEMAS since 2018, and she was responsible for really getting Cryo-EM for biological studies up and running. So looking forward to hearing how we can submit samples and learn more about the capabilities here at CEMAS.
Great. Thank you, Giovanna, for the introduction. Yes, so this is a talk that is mainly targeting potential new users of the Cryo-EM instrumentation here at CEMAS. So how you can get started, how you can have a successful project, how you can work with instrument managers and make them happy. And so we're putting all of this information together in this one talk. So hopefully it is also good for future users, and we can archive it and direct people here to this presentation. So I also wanted to make everyone aware of a couple of upcoming talks in this series. So the Beyond the Scope series includes not just Cryo-EM, but other electron microscopy related talks. So in June, we will have a guest talk from a visiting faculty member about some topic TM related. So that'll be announced in the coming weeks. And then we're taking a break in July for the M&M meeting. And then in August, we'll be back for an SEM talk given by one of our SEM instrument managers, Dan Beckdy. Okay, so you can see our website for more details about those talks. Oh, I'm sorry, I skipped a slide. Okay, so just in terms of CEMAS resources, obviously, if you're here, you notice that we have a monthly webinar series where we try and go into a little more in-depth about our instrumentation and capabilities. You're obviously also welcome to schedule one-on-one consultations with any of our instrument managers, and you can see all of the instruments that we have available. Then you see my email address there. Please reach out if you have any questions. Hopefully some of those will be answered in this presentation, but if anything specific to your project, please let us know and we can help direct you to the right person. And I also wanted to emphasize that our facility has returned to 100% occupancy and in-person training is available. So we've kind of sort of returned more to normal operations. So if you're interested in any of our instruments and want to use them in person, that's definitely possible. Okay, so just to give you a little more brief background about myself. So I was an undergrad chemistry major here at Ohio State. I moved after graduating from OSU to Caltech, where I did research more in sort of like nanomaterials, nanofabrication, where I did a lot of AFM and electrochemistry. So I left there with my master's and then finished my PhD at Emory University, where I kind of moved more into biological systems and looking at membrane protein activation of the Notch receptor and also doing fluorescence microscopy. So at that point, I was kind of hooked on biological questions specifically related to mechanosensation and mechanotransduction. So I joined the Sotomayor lab here at OSU, where I really kind of learned about structural biology from an X-ray crystallography aspect, but also doing molecular dynamics simulations, and then started transitioning into Cryo-EM. Which kind of is what led me here to CEMAS. And so as Giovanna mentioned, I was sort of here close to the beginning of when these two new microscopes that we have appeared and kind of building up the facility for Cryo-EM users here at CEMAS. Okay, so obviously there's quite a few instruments housed in this facility. They all sort of meet or exceed the manufacturer's specifications. So we have a great custom designed facility out here on West Campus. And I'm just going to be specifically talking about the Glacios, which is our 200 kV Cryo-EM, and then our Krios, which is our 300 kV CS corrected Cryo-TEM. We also have a Vitrobot, which is not listed here, but that is for plunge freezing of samples. So you see we have a wide array of instrumentation, and some of these might be of interest to those of you working with cells or tissues and have a Cryo-EM project there. Okay, so just to kind of give you some faces to go with the names. So this is sort of our core Cryo-EM team in terms of instrument managers. I tried to specify which instruments we work with in kind of our area of expertise a little bit more. So I'm on the left there, and I mainly work with setting up single particle data collections on the Krios. I also help with people that are screening on the Glacios. And mainly, I would say I try and kind of do more of the data analysis side of things. So trying to keep up to speed with what's going on in terms of processing data. Giovanna is our newest staff member. She came to us from the NIH and worked first around Subramanian. And so she has a lot of expertise with sample prep. So particularly using the VitraBot, vitrifying samples, also working not just with single particle type of samples, but cells as well. And she also has an interest in CLEM as well. And then finally, Binbin on the right, she is most experienced with electron tomography. So if you have a tomography project that you're interested in, I would definitely contact her. And then she also does help with screening and data collection on the Krios and Glacios. And so I wanted to bring this up because I highly recommend, if you have a project in mind, that you contact any of the instrument managers, but maybe specifically the one that would be have the most expertise in the area that you're considering your project in. So if it's single particle versus tomography, or maybe fib milling, you would pick the appropriate instrument manager, and then you can communicate with them directly. So we like to hear about exciting new projects, so please reach out to us directly. Even before starting, I highly recommend it. In addition, so this is a plug for our Cryo-EM interest group. So we have a very informal group of researchers across the university that have some interest in Cryo-EM. That doesn't mean that they are necessarily actively doing a Cryo-EM project, but maybe you're just interested in the technique or one of the techniques available with Cryo-EM, and you want to know more, or you have some questions about how to get started, you should definitely join our group. It's about, I would say, 30 to 35 individuals, but those are PIs, grad students, postdocs. And you can contact me directly to join the mailing list. But basically, we give an informal presentation that lasts about 30 minutes. And then we have sort of more of a discussion period about problems people are having with data analysis or sample prep. We cover sort of a wide variety of topics. We also have had outside speakers. So if someone has someone in mind that they would like to hear more about their work, we have invited outside researchers to give presentations, which is very convenient now because we just do it virtually. So we can have people from all across the globe give presentations. Okay, so mainly what I'll be addressing are questions related to, I would say, single particle analysis, but they can also be sort of, if you have more specific questions about working with cells or tissues or things like that, you'll probably have to contact us. But I kind of wanted to outline what are the things when you contact us, what you should tell us, right? So what do we as instrument managers need to know about your project? And I would say the first thing is you need to tell us, what are the primary goals of your research project and how can we help you achieve those goals? And then ideally, you need to have in mind what exactly you want to look at or observe. Is there a feature you're looking for? Is there, do you just want to see what certain particles, what their shape is? Do you just want images of that? Or is your ultimate goal really to do high resolution structure determination, which is single particle analysis? Or do you want to do a 3D reconstruction using tomography, anything like that? So you need to tell us what exactly you want to measure or observe. Also, a lot of times people come and they have seen something in a paper that they would like to reproduce for their own study. And it's very helpful when you can provide those references to us and tell us exactly what it is that you're trying to reproduce or replicate in your own study. So if you have references, please send those to us as well. So the last thing, which I think is kind of the most important in terms of being a user, is if this is a student or a postdoc or a trainee, do you want to be trained to do all of the experiments yourself so that you can operate things independently? Or do you want a staff member to complete the work in sort of a timely manner, like if you have a strict deadline or something like that? So that will kind of determine whether or not you should sign up to be a CEMAS user, or whether you just want to use us more as like a service-based facility. So you should decide before contacting us kind of what your intention is there. So I'm just going to go ahead and assume that some of you are interested in becoming a CEMAS user. So you would like to operate the instrumentation on your own, you like to make your own grids, that sort of thing. So I'm going off that assumption. So the following slides will kind of address how that process works. So I just want to outline it so it's clear to everyone what the steps are. Okay. Okay, so on the website, if you just go to our cemas.osu.edu, on the main page, there is a direct link. There's a box there that says becoming a CEMAS user, but there's also the full link here listed out. So you need to go to that page, and basically it outlines step by step what you need to do to access our facility. And as I mentioned, you have to provide details about your project specifically. You also need to complete all safety training before you can come and use any of our instrumentation. That's very important to us. We use FOM as our scheduling and billing system. So you do have to have an active account in FOM that is tied to a workday requisition. So this is a relatively new billing process that most of that people at OSU have gone through over the last few months or so. But our admin team has put together very helpful step-by-step instructions for how you can set up your requisition. And as instrument managers, we would highly recommend that you put enough funds into your requisition to cover the full cost of your experiments. And our rates are also on our website, but if you have any questions about how to estimate the amount you need on your requisition, please discuss that with an instrument manager, because we can give you some good feedback of what that might be. So once you kind of go through all these administrative check marks, then you can finally request training from an instrument manager to use a specific piece of equipment. So as I mentioned, Giovanna or myself can help with Vitrobot training. Any of us can do the Glacios training as well as the Cryos training as well. So please just make sure you contact the right instrument manager for what you're looking for. Okay, so that's great. Let's say you have everything ready to go. You have all this administrative stuff taken care of. Now you can actually focus on the science side of things, right? So like I said, my area of expertise is more in single particle analysis. So I'm going to talk specifically about samples that are coming to us for single particle analysis. So please keep that in mind. If you have some other type of sample, then we can discuss what the requirements for that would be. So everything starts on the user end, right? So you are going to come to us with your sample. And as a single particle sample, we would like to see certain sort of benchmarks. So we really like to see good biochemistry done on your end before coming to us. And so here I've just included an example that I did in my postdoc, which is just, this is a 52 or 53 kilodalton protein. This is a size exclusion curve that shows you, it does have some aggregate here at around 7.8 ml, but then you have a nice clean product peak at 12 ml. And if you look at the gel shows clean bands in B5 through B1. So we want to see evidence that you have a very clean sample, that you know sort of the sequence information, the molecular weight. You have an idea of how this sample is going to behave, what buffers it prefers, and we want to see that it has it's very clean sample. Now, this is not the only way to demonstrate something like this. So it doesn't have to be size exclusion chromatography and a gel. We have seen other examples of using mass spectrometry or AFM or ultracentrifugation. Those are all totally acceptable. It's just showing to us that the sample is very clean and the biochemistry is good. And so that will help lead to success kind of further down the road. And I want to mention at this point, so the biochemistry is more on the user end. So we understand it takes a very long time to get a nice pure sample, express it, and get a good yield. So we want to make sure that we do everything we can to help you with that precious sample. And that being said, so everything that we do here, which would include negative staining, preparing cryo-EM grids, and then screening them, we have training resources on our wiki, which I don't know if we've advertised as well as we should have, but the link is right here at this u.osu address. You do need OSU credentials, but we have uploaded both written protocols and training videos that show you step-by-step how to do everything. So everything that you really need for single particle analysis. So I highly recommend going to this website also after you've made your sample, or maybe simultaneously before you come to us for training, because this will make things go in-person training. It'll make things go much smoother. So you'll be prepared if you kind of preview these videos and the documentation before you get here. Okay, so for negative stain, like I said, we do have videos about how to do negative stain and sort of more like instructional, like specific protocols, how to do this. But our recommendation is always to use fresh sample that has never been frozen. Ideally, you would run the gel and you would see it's pure, and then you would immediately move to negative stain. It helps if you know the concentration of your sample pretty accurately. If not, we kind of have to do a little more guesswork about how to properly dilute it to see the right density, particle density on the grid. So just make sure to try and measure as accurately as possible. And it really is a key step in understanding your sample homogeneity and quality. So I'm showing you two images here of two different types of particles that would be sort of approved for moving on to making cryo-EM grids. What we like to see is essentially no aggregation or very little aggregation, nice even staining. You'll notice that in some of these images, you can see almost some like finer texture within the structure itself. You will get many more particles, so they're much more densely packed in negative stain than they would be when you make the cryo grids. So that's just something to keep in mind and why it's important to know the concentration. And I know Giovanna's a fan of this, but you can actually select these particles and create a low resolution model if you particle pick enough of these. So it can provide quite a bit of information on its own before moving to cryo-EM. But this is kind of one of those key steps that people will maybe try to skip over, but we don't recommend skipping over negative stain. Okay. And then once we see that the sample looks good in negative stain, you would move on to actually prepping cryo-EM grids. And so, as I mentioned, we have a plunging system available. It's a VitraBot. It's free for our users, but you do have to be trained to use it. And it probably takes, I would say, 2 to 3 sessions to get approved to use that on your own. It's more about grid handling, and it's a little tricky once you get liquid nitrogen in there. And so if you're totally new to cryo-EM, it does take a few sessions, I would say, to really get comfortable using the VitraBot and plunge freezing your own grids. We do provide sort of common grid types available for cost. So we'll just charge you whatever we purchase them for, but we have quantifoils and C-flats, also lacy carbon grids available. So all those common grid types we have here. And so maybe once you find a grid that works well for your sample, then you could order your own batch. And the whole goal of this process, right, of making cryo-EM grids is that you want to achieve very thin vitreous ice. You don't want to see crystalline ice contamination at all or very little on your grid. Just like in the negative stain, you want very good particle distribution in the holes. And you want to see a lot of grid squares that can be used for data collection. So if you only have one good grid square on an entire grid, that's not going to be enough data for you to successfully do a reconstruction. So we want to see, at least a handful, like 5 to 10 grids that we could see collecting data from. And as instrument managers, we highly recommend taking what's called an atlas for any grids that you think are promising and that you might consider using for data collection. Okay, so I'll show you on the next slide. Let me just show you what I'm talking about. So, okay, so the grid screening process we do on the Glacios generally, so the 200 kV instrument. So the process is pretty straightforward. You go from low mag all the way up to your data acquisition magnification. The atlas here that you see on the left shows you what the entire grid looks like. So the diameter here is about 3 millimeters. And ideally, the reason why we ask you to take an atlas if you think it's a promising grid is because the atlas is almost like a fingerprint of that particular grid. So if you look at it, you can see, okay, here there's like 3 broken squares in the center. Oh, sorry, you can't see my mouse. Let's see if I can make it. Okay. So here in the center, right, you can see there's these 3 broken squares. There's a some pooled thick ice down here. So it becomes very identifiable to know which grid is which. So that's why we highly recommend the atlas. The atlas is not just one image, it's a tiled image. So this, I believe, is 16 images that are stitched together. So that's how you can get this giant view of the entire grid. And then this way, if we move into, let's say, move this grid into the Krios, we would know exactly that it's the exact same grid that we looked at. So then you move up to higher magnification, right? So you want to look at an individual grid square. So that's what you see here. This is in picture number 2. So slightly higher magnification. This grid square is very nice, has a lot of thin ice on it. You do see some small ice crystals here, which are these dark features, but that's okay. We can exclude those holes from data collection. And then at the next highest magnification, which is called hole eucentric, you can see here an individual hole, and over the hole is vitreous ice. And then if you were to take a single snapshot in the center of the hole here at the highest magnification, which is data acquisition, then you can actually see individual particles inside of the hole. And you want to see nice particle distribution, no aggregation, same things as you would with negative stain. Okay, so hopefully this gives you an idea of how the screening process would work. And so as I was saying, this is an example of an excellent grid for data collection, because there are many squares here that are usable for data collection. So there's probably over 20 grid squares that could be used. So this is kind of what we would like to see in an ideal grid. Okay, so after screening, let's say you want to save your samples, right? So maybe you're waiting to get the perfect grid, which is totally fine. We understand that your resources are very precious. So you can store these grids for in the long term. So we have a large storage doer here where you can move the samples into these sort of multicolored storage boxes. So keep in mind that unlike a traditional TEM grid, these grids have to be clipped. So you can see a picture of like the front and the back of the grid. So there's actually a cartridge essentially that is like a sandwich around your grid. And that's so we can use the autoloader system on the Glacios and the Krios to essentially grip the samples. So those clipped grids need to be stored in special boxes, which you can see here. Those we're recommending that users provide their own boxes just because they may be stored for long periods of time. It could be months, it could be years. But we unfortunately are unable, we cannot track the details for all your samples because we can add up very quickly. So we highly recommend that each group has, a very consistent naming system so they know which samples are located where. These are all available from commercial sources, so we can provide you details for that. We also need to know if there are any requirements for storing your samples for any period of time. So in some cases, funding agencies might require that your samples be available for a certain amount of time. If that is the case, please let us know so that we can make sure that that's taken care of appropriately. We give each group basically a 50 ml conical that can hold many boxes like this. And so we ask users to kind of periodically go through those and make sure that, they're only keeping the ones that are important for their research. Okay, but I think that's hopefully that's pretty standard for even your own samples in other cases in your own lab. So please keep careful track of that. Okay, and not the last thing, but in terms of data analysis. So let's say you've gone through this whole process. You have a beautiful grid, you decide you want to collect data, and the data will be collected relatively quickly over the course of a few days, let's say. And so then you'll be left with lots and lots of images of your single particles, or it could be tomography data as well. And if you're doing screening, you might just take individual snapshots of your grids. And those you can save onto your instrument drive here at CEMAS. And so if you need your own instrument drive folder, which I would recommend all new users get, you can ask us to have that set up for you. Then you can actually access the instrument drive folder through this website here. And that works well for very small snapshots or images, individual images of grids. But once you get to the point that you actually have done a data collection, you're probably left with terabytes of data. So let's say a couple terabytes of data. That's not something that we can put on our instrument drive for you to access through the web. That's just too much data. And so our preferred method, I would say, of data transfer right now is to use Globus. So we essentially would need you to have an endpoint where we can tell your data to go there, and then it will just transfer without the use of an external hard drive. So we just do that kind of through the web or through the internet, essentially through a fast internet connection. We can still do external hard drives as a backup, and I know some labs prefer that. It'll be a little bit slower, right? Because we have to copy it multiple times onto your drive, but that's also an option as well. I would say, so we do have some ability to do data processing here at our facility, but we would highly encourage, but not enough resources, unfortunately, for everyone to use. So we mainly try and keep it to for our internal staff use, but I would highly recommend that if you're new to single particle analysis, that you reach out to collaborators, or there are other ways of doing this. You can also, if you're very invested in Cryo-EM, I highly recommend looking into purchasing your own computational resources, or as a standalone workstation, or using, for example, apologies, that's not my phone, or using the Ohio Supercomputing Center or something like AWS. So those are all good options for data processing. So if you're not an expert in data analysis, I would say reach out to someone who is an expert and ask them what the best way of moving forward is, right? So unfortunately, we do not have the staff resources to help everyone with their data processing as well, but we can sort of help you, guide you in the right direction. Okay, and then lastly, just in terms of, as I mentioned, we do deal with a lot of data. So our Cryos can produce probably, several terabytes of data per day. So if it's running around the clock, you can see how quickly we're producing data. So that being said, we can't store data indefinitely, especially at that rate. And so we have sort of some guidelines about how long we can keep data. So just to be upfront about how long we can keep everything, we generally store on our local instrument, like on the microscope, let's say server, we can only keep that for up to 48 hours maximum. And then we copy that over onto our college instrument share, where it can stay for up to 2 weeks, but we can also extend that if needed. During that 2 weeks, at some point, you would either transfer it onto an external hard drive, or through Globus, we would transfer it to your group directly. So unfortunately, we cannot provide long-term storage of data. Like I said, it's so much data that it's just not feasible. And then after those time limits have been exceeded, we can't guarantee that we will have that data available. So it may be removed if we need to make space on our instruments. So that's just to be upfront about data storage. Okay, so that kind of takes you from sample prep, all the way through to how you treat your data. So I'm just going to go back and put this slide up real quick, just to emphasize that, if you're starting your project, you probably have a lot of questions. And I would say, I'm putting this contact info up here so you can write it down. Please reach out to us if you have any questions about the entire process. We want obviously your project to be as successful as possible. And I tried to put in this presentation things that would help make it go as smoothly as possible. But really, we would love to hear from you. I talked about single particle samples, but if you have samples that you're interested in doing, for example, cryo-FIB or micro-ED, those are also techniques that we have available here. So please reach out and we will put you in contact with the right people. Okay. Again, just wanted to highlight the upcoming events. So we will have a talk in June and August, but we'll be skipping the month of July for M&M. And this last slide here is how you can reach CEMAS in general. So we're lots of places on social media. So please reach out to us in any of those ways as well. Okay, that's all I had for my presentation. And if you have any questions, please put them in the Q&A. We'll hang around for a few minutes to see if anything pops up. Or if you want to talk about specific needs you might have, just throw it in there, that's fine. I don't know if anybody else, moderators, anything else you want to add?
No, thanks for a great talk.
I hope I covered things that, will make your instrument manager happy.
Yeah,